All right, I think it's time to get going. I wanted to just do a quick introduction of what brings this event to campus before introducing our speaker, and then I plan to give him the lion's share of the time, and also hopefully have some time for questions afterwards. For those of you who've not met me, I'm Travis Cram. I volunteer with TU. I'm also a professor here at Western, and I also direct the debate team. So what is it that brings North Sound, Trout Unlimited, and the Western debate team to co-sponsor an event besides me? There is a tie there beyond just convenience. One key aspect of debate that I think is really admirable is its capacity to dig into big ideas, put ideas into tension, find what is the best way to express the best solution for many of the problems our world is facing. So throughout the year, my students have been reading original peer-reviewed research on climate policy and what should be the federal government's kind of trajectory going forward. And I asked them if they are interested in sponsoring this event, even though it's not a climate change focused event, it's certainly caught up in it. And their commitment to trying to explore big ideas and find the best way to advocate for better solutions drew them to this event. And for North Sound TU in particular, the reason why we're focusing on the Skagit specifically, beyond the fact that it's a river we all love, it's a river we love to spend our time on, it's at a delicate point. We're at the very final stages of a 12-year moratorium on hatchery plants in the river. There's increasingly looking like there will be fish passage that's added to Seattle City Lights dams on the Skagit, and a lot is in flux. And that's to say nothing of what's in flux politically at the federal level in this country. So at a time of a lack of clarity, it's a good to look for big ideas. And we turn to one of our best friends here at North Sound, Nick Chambers, who's been working on wrapping up his master's in science with the University of Washington's fisheries science program. He's been collecting data on steelhead, especially different life stages in the Skagit Basin. And when he sent me the proposed title of his talk, I was like, that's a big idea. What do we do going forward, recognizing in a lot of ways we've blown the world of steelhead up? And so with that, I just want to turn it over to Nick, and looking forward to hearing what you have to say.
Thank you, Travis. Bear with me one second here. All right, can you guys hear me okay? All right, so just a quick introduction. I see a few faces out there I don't recognize. I grew up in Southwest Oregon. I was born in the mid-'80s and grew up fishing from a very young age, and I was just old enough to really be aware of all of the ESA listings that started occurring in the early 90s, right, when we first had some of the biggest, really big recent collapses in returns of anadromous fish, from steelhead particularly in the Columbia River Basin. And so, growing up, it was always a story of, man, you should have been here yesterday. It was kind of a sad story, honestly, of, you'd see a lot of pictures, you go into a tackle shop, and a lot of those fisheries just don't exist anymore. And so that really led me to get a degree in fisheries and kind of go down this path I have. And so June of 2025 is actually 20 years since my first fisheries job, so I've been doing this a while. It's been kind of a circuitous route to get here, so I'll spare you the details, but worked for Trout Unlimited for 4 years and got to know a lot of the folks here, and that's kind of how I got connected with this group of people, and they've been a huge help to me in getting accomplishing this work that I did for my master's that I'm going to talk about a little bit today, and just wrapping that up at University of Washington. And all that work was on the Skagit River, studying steelhead, and in particular studying fry, the early life of steelhead and what happens to them during this first summer of life. And let's see, maybe. Can I just? Oh, there it goes. Yeah, it's working now. Okay, problem solved. So, steelhead are what we would call a generalist. They occupy the broadest range of habitats of any of our Pacific salmonids. They currently exist all the way from the mountains of Mexico all the way up to the Kuskokwim River in Alaska. They have the broadest entry timing of any salmonid. They can enter freshwater 12 months out of the year, and they can even spawn between December and July. And in places like the Klamath, where we have large populations of resident fish, we actually see them spawning 12 months out of the year as well. And so they have this really broad array of life histories that allows them to use a really broad suite of habitats, right? All the way from lower mainstem rivers, where they most closely overlap with Chinook, all the way to tiny headwater creeks, where they really overlap with coho and things like coastal cutthroat trout. And so there's some important biological differences between them and their salmon cousins that we don't necessarily always account for in management very well. And while they're not currently commercially valuable, historically, they actually they were quite valuable. And so, unfortunately, like their salmon cousins, most of the populations that we have, particularly in the lower 48, are at a small fraction of their historical abundance. And so likely everyone's heard of the 4 H's. These are habitat, hatcheries, harvest, and hydropower. These are some of the main reasons for decline, right? I think a lot of us are pretty familiar with that. So this image is from the Klamath River, probably in the 1930s, where they would gold dredge and they would dig up the whole river valley bottom and put it into these big tailing piles. And most of these tailing piles still exist. You can drive down the Trinity and some of these places and see these. And then the river just gets channelized against one bank of the valley wall, right? And then between logging and splash damming and all these different impacts that we've had, we've really destroyed their habitat in many places. And then on top of that, we also harvested them at very high rates. So historically, they were actually one of the most valuable species in Puget Sound because there was a pretty large fresh fish market, and they would put fish on trains and ship them around the West, and Steelhead actually kept better than the salmon that they would put on there when they would salt it and put it on trucks full of ice. And it also came back at a time of year when none of the salmon were returning. So we actually harvested them at very high rates. And while we don't know exactly how many fish were returning to Puget Sound, we can actually use some of these commercial cannery records to come up with historic run size estimates. So when Puget Sound steelhead were listed in 2007, Jeff Hart at NOAA came up with some estimates that put the number of steelhead returning to Puget Sound somewhere in the 350 to 500,000 range. And that may even be an underestimate based on the size of fish he estimated. It's a guess, but that it could be even more. And we don't know how many are coming back to the Skagit, but if we just say the Skagit's the largest river in Puget Sound and kind of divide that out by basin area, we may have had as many as 100 to 200,000 fish coming back to the Skagit. Currently we're averaging roughly 4,000 to 6,000 over the last few years. So tremendous decline in the number of fish coming back. But so have we really destroyed all the habitat that's left so that steelhead are just in this like post-apocalyptic position right now, right? That's what we hear from a lot of people, right? It's maybe not even 4 H's. You'll hear people just say habitat, 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 right? The only way that we can recover fish is to restore habitat because we've destroyed it, and it's just, there's not enough left. What we have right now is all that we can get. And that makes sense in some ways, right? But if we look at some of these examples we have, like the Columbia River Basin, where we spent $15 billion in the last 25 years or so, wild fish have continued to decline. So not only have they not responded, it hasn't really even stemmed the decline. And this is what we see broadly, is we're not really seeing a measurable benefit to all this habitat restoration work that we're doing. And so that's kind of a puzzling result in some places too, because for one, we know that habitat's important, right? I don't mean to downplay that. Fish need cold water, they need cover, they need all these things. So restoring habitat and improving it should provide a benefit, but we also have relatively pristine habitat in some places. So if we think about the Middle Fork salmon, for example, it's almost entirely within a wilderness area. It's maybe not totally pristine, but it hasn't changed that much from historical condition. And both Chinook and Steelhead are really struggling, and in some years barely even returning. And so the question is habitat truly limiting? And how do we even measure that? How do we determine what is the relationship, what is the status of the population in relation to the habitat that remains? And so one of the ways that we look for this is what's called density dependence. And of course, scientists like any specialized trade, like to come up with fancy names for pretty simple concepts. And all this means is that as the density of individuals increases, there's more competition. And so either they grow slower, they don't survive as well, or they're gonna move out from certain areas and they're gonna disperse, right? They're gonna find a lower density area. And so when we look for that, what we see is that it's ubiquitous. And so that suggests that, well, these fish are fully occupying the habitat that's there, right? If there was a bunch of really good habitat and the fish were really at low densities like we see, and the quality of habitat was really good, we wouldn't see density dependence, right? They would all just survive and grow really well. And that's not necessarily what we see. And even in the Skagit, this is from a recent paper from Mark Sherrill at University of Washington, and this was sort of asking that question, do we see density dependence in the Skagit? And so what that really looks like here is this x-axis here is spawners, so that's the number of fish that come back and spawn. And then the y-axis is recruits, which all that is how many offspring did they produce in the next generation, right? So you just have number of adults in one generation to number of adults in the next. And what we see is, for one, this looks like a shotgun cloud of points, and we'll touch on that later. It doesn't really look like there's a relationship there to me, but when you fit a model to it, it tells us that as we get more spawners back, at a certain point, we don't get any more adults back, right? That levels off. And so why that's occurring is when we're down here, we have what we would say like compensatory survival, which is essentially density is really low, everyone survives. So if you're a millennial like me, I don't see too many in the audience here, but we're the generation where everybody gets a participation trophy, right? Like that's pretty much what's happening down here is this, oops. This is the participation trophy zone where everybody gets a trophy, everybody survives. You all get a rock with plenty of food to hide behind, and everything is gonna be great. And then as you start to get up further on that curve, then there's a lot of competition, right? Presumably this would be older generations that had it harder than us, I guess, you know? And so there's tremendous competition up there that prevents them from being more abundant. And what that leads us to is an estimate of capacity for how many fish we can fit into the watershed. And so on the right over here, estimated carrying capacity from this model was just shy of about 8,000 fish for the entire Skagit Basin for winter steelhead. And with a range of about 6,000 to 12,000, so we're pretty certain it's within that range. And if you look at where all these curves level off, it's kind of in that range, right? So where they plateau, that's where we say that would be capacity. That's how many fish we can fit into the watershed before we just don't see any more productivity coming out of that. So far below what those historical estimates were. And so where is density dependence really occurring? One thing that we don't do very well in the Pacific Northwest is actually monitor juvenile steelhead. We tend to do things like pit tag fish, but we only pit tag larger fish. They're like what you would put in your pet to track them, right? And you scan them and you get a number back. And so we have to have fairly large fish, usually at least a year old, to track. And then the other way we track fish is through smolt traps. And that works relatively well for things like Chinook or pinks that are going out as fry when they're really small. But steelhead are going out when they're 2, 3 years old, so they're really good swimmers. They're basically trout at this point, and so they just avoid the trap, so we don't catch very many of them in most places. So we don't really have a good picture of what's going on for juvenile steelhead. But steelhead are actually more similar in many ways to brown trout and Atlantic salmon than they are to our Pacific salmon, in that they have they're iteroparous for one, so they don't die after spawning. They have both a resident and anadromous form that spawn together as one population. They tend not to disperse from reds early in life as juveniles, at least for Atlantic salmon and brown trout. And that leads to a scenario where we have really intense competition early in life near reds. And so density dependence is actually strongest, what we see is early in life during this first 2 to 3 months after emergence, right? And that's really dictating what occurs in that curve we saw earlier, right? So it's not that, we assume that it's just somewhere during this freshwater life history of 2 to 3 years where juvenile steelhead in the river, there's this density dependence. And what the literature is actually suggesting that this is really occurring likely during this first summer of life, during this very short window. And there is some evidence of this for steelhead, although this largely comes from the Atlantic salmon world. And that's essentially just looks like this plot on the right here. The more fish you have, the more fish you start out with, the higher your mortality rate is, right? So this is essentially what we see when we look. But what's notable here is that this is occurring during the first year of life rather than later life stages. And why this happens is their fry, right after they emerge from the red, essentially undergo this very rapid pace of life, right? They have to emerge, and there's no one to teach them how to feed. There's no one to teach them where to go. So it takes them about a week for all the fry to emerge from the red. Within a couple days, they start already competing with each other. They're nipping, chasing each other. They're establishing little tiny territories behind rocks. And that sort of forces later emerging fish progressively further downstream. And that forms what we would call a dispersal kernel. And this appears to happen within the first 1 to 2 weeks. There's this pulse of emergence. They move out over a couple hundred meters, and then they likely sit there throughout the first part of the summer. And then they only have, particularly if you think about fish in the Skagit, they may not emerge from the gravel until June or July, and they really only have until September before fall rains may start to really grow and achieve a substantial size that's gonna allow them to survive through that first winter. And if they're only get to be about 36 millimeters in September, which we actually saw a bunch of those, it's pretty unlikely they're gonna survive. And if they do, they're actually likely to be an older smolt. So it's not that they can achieve catch-up growth during the next couple of years in freshwater, it's that actually sets them on a trajectory that carries through to the smolt stage. So what happens during this first summer of life really carries through all the way to the smolt stage, even for 3, 4, and 5-year-old smolts. And so it really is actually quite a very important stage, but it's one that we don't necessarily look at very much. And so I talk about there's not very many studies that look at how far these fish disperse early in life, and they're almost all from hatchery fish, where we raise fish in the hatchery for a week or two, sometimes a couple months, and so we're feeding them, right? And then we dump them into the river. And so it's hard to know, is that what happens when they emerge from a red or not? And for Atlantic salmon, what we see is the vast majority of these fry stay within about 200 meters of the red, which is quite the opposite of what salmon do, where they may move multiple kilometers. There was a paper just done on spring Chinook where the average dispersal distance was about 3 kilometers. So if you think about that, they're able to move and access a bunch of different habitat within this early period of life, where steelhead appear to be staying close to the red. So the black bars here are how far fry disperse from reds. So you can see most of them are staying within about 200 meters of the red. A few of them go 400 up to a kilometer, but not very many. And then interesting, the hash bars and the white bars are 1 and 2-year-old par. And they're also staying very, very close to the red. We really don't see, as long as conditions are reasonably good, they have things like water and food, they really stay oftentimes in the same habitat unit their entire freshwater residence before becoming a smolt and actually going to the ocean. And so remember that curve we saw earlier? This is what we see. This is just the first 2 months of life. This is the first summer of life, right? We already established this pattern of density dependence. And then really it appears to carry through to later life stages. And then by the time we get to the adult stage, this is from that same paper. This is on brown trout. And it starts to really look like the Skagit, right? There's really not a clear relationship here. It looks like somebody's patterning a shotgun or something, And so this is a real phenomenon, but it becomes very difficult to detect when all you look at is older life stages and we're not looking at fry. And so this brings me to my research. And so my main research question is pretty simple, which is just how far do fry move from reds early in life? And then really the pattern, if our prediction is accurate that they're not moving far from reds, then really where the fry are at is where the adults are going to be, right? So the distribution of spawners is really going to matter and tell us about where the fry are going to be. And so does the clustering and distribution of spawners influence change with abundance? And then does this lead to the potential for density dependence in rivers like the Skagit? Are we seeing density dependence occurring at the fry stages? That's what's driving this relationship that we see at the adult stage. And so all my work was in the upper Skagit River. So Seattle City Light was one of my funders. And they were very generous to share all of their GPS located red data that they maintain between the mouth of the Sauk River and Shovel Spur Rapid, which is 31.5 kilometers upstream. And then we did sample some reds up closer to the dam. But most of the work was in this 31 kilometer reach in the lower river where we have 10 years of GPS located red count data. And so to track dispersal, essentially what we did was go out in the spring, and I piggybacked on Seattle City Light and did extra red counts to make sure that we identified every single red. And so I would go out and find single isolated reds that didn't have a red within another kilometer or so. And then, so this, this red oval here would be a red, and then the blue dots would be transects that we would snorkel. So we'd lay out transects 10 meters downstream of the red, up to about 300 meters, and then snorkel upstream and count all the fry within each transect. So we would get a pattern of fry along the river channel. And this actually works quite well. You can see the orange arrow there is pointing to a snorkeler, so it gives you some scope of the scale that what we're sampling here, but fry largely stay within about a meter of the shore during this early period of life. So we're out there trying to do this within the first 1 to 2 months after emergence. And so this is what we found. This is an example of what we call dispersal kernels, which is just the shape of the distribution of fry counts from the red. So the reds are red triangles there, and then the gray bars are just the observed fry in the downstream direction from the red. So you can see the vast majority of fish are staying within 200 meters, sometimes up to 400 meters from the red. But so this aligns pretty much exactly with what we would expect to see from Atlantic salmon and what we've also seen in some of this work that uses hatchery fry. And so once we have that, then we could basically just, this is just a dispersal kernel. It's a statistical distribution. It's kind of boring, but essentially once we fit a distribution to all our dispersal kernels, so we could describe the mean and the spread, right? So the average fry was moving 145 meters from the red, and then 95% of the fry were expected to be within 312 meters of the red, right? And so just describing how far they're moving from the red allowed us to do some cool analysis. So this is just an example of what these GPS patterns of reds look like. So 93 was the lowest number of reds we had in this reach, which is pretty low for 30 kilometers of river. And the highest year was 302. And so what we did was take that dispersal kernel and apply it to every single red. And where we have overlapping reds, we would add up all the area under the curve, right? We scaled each dispersal kernel to be the same size. And this is what it looks like when you look at that, right? So we had 10 years of data. Each one of these is the predicted distribution of fry along the river channel, right? So when you have these big, tall blue bars, that's a really dense cluster of fry. And you notice that as we go up in, right, so we have low numbers of reds through here, and then going up, we tend to get fish spread out more, and also they get more dense. And so what that looks like is on the left here, we have the total number of reds in the reach, and then on the right is how much distance is actually accessible to fry in any given year, right? So we see at the lowest number of reds, we only have about 10 kilometers of habitat accessible. And in the highest years, we have about 18 kilometers, 19 kilometers. So we're almost doubling the area that's actually accessible to fry. So this is the area that's actually being used by fry early in life, right? So it's not consistent. So what we're seeing is as we get more fish back, they're expanding and they're occupying new habitats, right? They're not just necessarily going in on top of each other. And then on the right, it's a little confusing, but basically, if the fish were filling up the habitat perfectly and evenly, as much as possible, they would follow that blue line. And what we're seeing is that they're actually up here on this red line, right? So they're much more clustered than we would expect if they were perfectly uniform. And if we think about back to that density dependence curve, what we're assuming down in the participation trophy zone is that these fish have equal access to all the habitat in the watershed, right? So we're assuming that they're a lot less densely packed in these low abundance years than they are, right? They're not actually filling up the habitat in sort of an ideal way. They're remaining clustered at low abundance. And so I was talking about where the reds are dictates where the fry are. So looking at how fish actually fill up the habitat, what this is we have paired years. So X and Y axis are both year, and then there's an NA column running diagonally. So each square in there is a pair of years, and then the NA is when you have the same year matched to itself, right? And so the brighter colors, the reds and yellows, we have a higher proportion of areas that are reused for spawning, right? So we took that river network and we cut it up into 100-meter bins. And every time that we had the same 100-meter bin with a red in it as another year, we would count that as a proportion reused. So we see in the lowest years, 25% to maybe a third of those bins are reused for spawning. And in the highest years, we're reusing 2/3 of those sites. Over 65% of those are reused. So fish are reusing the same spawning locations year over year. And if you notice that on that diagonal, the colors tend to be pretty bright. So fish are using the same sites year over year. And in years that are further apart, 5, 10 years apart, they're not using those sites as much. So this is likely because we have overlapping age classes and these fish are related to each other and they're actually homing back to some of the same sections river and then selecting similar spawning sites. We can't say that for sure, but that's one thing that we think is occurring. And this is important because, and I'll get into it here. So we took all that fry data and where we cut the network up into the river network up into 100-meter bins for the red counts, we did it with 10-meter bins for the fry density counts because that's how big our snorkels transects were, what we actually surveyed and counted red. So imagine taking that whole river network up, cutting it into 10-meter bins, and you have an estimated density for each 10-meter bin. So we have a whole bunch of numbers, and then we divide that up into 4 quarters, right? So we have a high-density bin, a low-density bin, and then 2 in the middle. And so this is the number of fry that are in each bin based on the number of eggs that were deposited, right? So this is assuming like 100% egg survival, 100% to emergence, which isn't realistic, but it gives us a potential for what there could be in any of these places. And so what we see is that in these low density bins, we don't really see a whole lot of change in density, right? And so if you're gonna have density dependence, if you're gonna see that curve leveling off, you need a change in density because you need that change in the level of competition that's occurring. But in these high density bins, we have a very marked change in density, right? So that's evidence that density dependence is occurring. And what the colored dots mean is the darker colors and the larger dots mean that there's a higher proportion of reds in core areas or in highly reused areas, right? So what this is telling us is that in low abundance years, fish are retreating into core areas. So they're coming back to those presumably the best habitat patches, and we only have them occupying the best habitat patches, and as we escape more fish, then they're moving out into satellite patches, right? And so what's happening is we have, in core areas, we have really strongly increasing density because those are really good habitats and they're producing a lot of smolts. Fish are coming back to those places in high numbers, but then we're also expanding out, right? So we have density dependence likely occurring in, what's driving it is these core areas. And what you don't see on here is the number of bins in each category is not the same, the number of 10-meter bins. These fish are only in the low abundance years, they're only about 400 meters of habitat, and the highest years are in less than 2 kilometers of habitat in that highest density bin, right? And so this is a tiny fraction of that entire reach that accounts for sometimes most of the fry that are produced in any given year. And so this is the same basic concept, but instead of a total number of fry, it's the percent in that quartile, right? So in low years with low red numbers, we have 40 to 80, or excuse me, yeah, about 60 to 80% of the potential fry production comes in these low density areas. And then in higher run size years, we have maybe 50, 60% of the fry are coming from high density bins, right? So this is just strong evidence for density dependence. So it's kind of confirms what that other paper was showing us. But again, it's telling us this is occurring on a really, really local scale versus this whole watershed scale. Where we traditionally aggregate the whole watershed together and look at these populations, the Sauk, the Skagit, the Nookachams, everything combined, this is telling us like at the scale of where it's happening. Okay. And so why does this matter? Well, so this is an example from the Rogue, which is where I grew up, so I'm partial to this. So the Rogue has 3 distinct sort of races of steelhead, 2 summer runs, one enters in the fall, one enters in the spring, and then you have a winter run. And so this paper's on this fall entering summer steelhead. And so all these little areas with thick lines are where these fall summer runs spawn. And if you notice, they're all disconnected from each other, right? They're not spawning in the main stem, that's just showing the river's larger. They're just all in these little tiny tributaries all over. And the reason they're only in those tributaries and not in the main stem is because all the main stem areas have winter steelhead spawning in them, right? So the fish are partitioning the habitat up into little tiny creeks that summer runs spawn in, and then winter runs are spawning in the creek that's right next to it that's just a little bit bigger. And this watershed is a little bit bigger than the Skagit, and as far as the anadromous zone, probably. It's a large watershed. And so why this is important is the way that they're splitting up the habitat, right? So summer runs spawn earlier, and they start spawning in December and January, and then it tapers off through February. And winter runs in this part of the watershed don't start spawning really until the end of February. So there's isolation by time where they don't interact, but then summer runs also specialize in spawning ephemeral streams here, right? So they're spawning in streams that average less than 35 CFS in the winter and then go dry between May and August. And so winter runs are spawning in the ephemeral streams that are just downstream. And then those juveniles have to get out of the watershed before it actually goes dry. And so steelhead, remember, they're very diverse. This is an example of how they're able to use these very different habitats by entering at a different time of year, spawning at a different time of year, and in a different place. So spawn timing is often correlated with a different spawning location. And we see this even within, so this is a study on winter steelhead within the Quileute. And so we look at the sole duck first. We have these 3 different reaches that are color-coded. And then this plot on the right shows the arrival date of spawners in that reach. And what we see is they're essentially filling the watershed from the top down, right? And it's arrival date, not spawning date, but spawning date is generally closely correlated with arrival. And so even within winter steelhead, and these reaches are relatively close to each other, we see difference in run timing that leads to a difference in spatial distribution of spawners, right? Early entering fish are using parts of the watershed that are a little bit further up, and then the very latest fish actually went all the way back to the top of the watershed. And so we have what we call spatial structure here, right? Because the fish are not just broadly mixing. A fish that comes in January is not necessarily the same as a fish that's going to come in May, and they're likely to spawn in a different part of the watershed. And then we have the same thing on the Kalawa here, right? Where even though these two reaches are right next to each other, over 4 or 5 years, I can't remember how many years of data there was here, on average, there was a difference in entry timing and arrival timing on this very fine scale. And so we don't know exactly how close fish home. We have some evidence from Idaho and Middle Fork salmon that 55% of female Chinook home to within 1 kilometer, right? So this is basically back to the same habitat patch they came from, and 87% home to within, or up to 10 kilometers, right? So if we think about my study reach, it's 30 kilometers. Most of the fish are homing within that reach. So they're not necessarily a fish that you catch at the bottom end versus a fish at the top end. Likely is not just randomly distributing itself throughout that reach and selecting the best patch of habitat. They're coming back to where they came from. And this makes sense because they don't necessarily know the quality of the habitat around them, right? The best information they have is that they survived coming from that place. And so they're likely to go back to that place because there's a good chance that it's a good place to grow up. And there's even some evidence that fish that stray as juveniles are more likely to stray as adults because presumably they had a crappy childhood, there wasn't enough food, whatever, they left and found somewhere else and they're like, I'm not going back there, I'm gonna take my chances and go somewhere random. And it makes sense. If we look at Atlantic salmon, migratory birds, a lot of species that home, that may go on these long migrations and come back, there's usually a fitness advantage to coming back to a pretty local place. And fitness in this case is just the number of offspring you produce, right? You're more likely to have successful offspring if you home back to a local scale. So we can't assume that fish are just randomly coming back to the watershed to spawn. And so what does this all mean? Well, if you have limited dispersal of fry early in life, you have a really high mortality rate that's proportional to density, right? So the more fish you have in a local area, the lower your survival rate is. Then you're likely to have strong density dependence early in life, and that life stage is really going to present a bottleneck to productivity, or it potentially can, right? Because fry can't access all of the habitat. So we're assuming that fry have access to all the habitat in all of our models where we estimate productivity. We're assuming that they can access everything and that they also have knowledge about the quality of habitat, so they're always going back to the best places, which is unlikely to be true. And par are more mobile, so they could access some of these other places, but the greatest period of mortality in life has already occurred prior to that par stage, so they're unlikely to overcome what is set at the fry stage, and that's what we see. And so what this means is really that density dependence, where we're interpreting it as a signal that the habitat is fully saturated, right? It could be simply because we have local clustering in a watershed, right? So even as populations contract, they retreat into core areas, and they stay more densely packed than we would assume. And we're not accounting for that in any way in our management. So if you think about, let's say, an escapement goal, we had the quill earlier. So we go out for a couple years, we count how many fish we have, and then we set an escapement goal based on that. And then for 40 years, we harvest fish down to somewhere near that escapement goal. We're very likely to still see density dependence when we look at that population, right? Because they're still clustering in space and time, regardless of how much habitat is available to them. And so this plot is essentially, if you look at this upper line here, it's essentially a stock recruit curve. This is just a whole bunch of those overlaid into like a 3D plane, right? So if you just start here and you keep your population there, you're never going to know that there's more habitat that could be suitable that's not being used, right? And that is one thing that we could be doing in some places. And similarly in the Skagit, we have a tiered harvest rate, right? We don't have an escapement goal. But we also don't know, what we see is a linear increase in that accessible area to fry. Well, when they've looked at that in Atlantic salmon, a lot of places, they actually see that taper off, just like you would here, because they actually fill up the habitat, and then they start having to pack in on top of each other. And we only have 1 point at the upper end of that. So it's possible that, that is the limit, and we just don't see that relationship. But that's up around 8,000 fish, where if we only have 5,000 or 6,000 fish come back, is that truly enough to fill up all the habitat? That's the question, right? How many fish do you need to escape to really actually use up all the habitat spatially? And that's something that we don't account for in any of our fishery management plans, is how fish are actually filling up the habitat and spatially structured. And one reason that we likely see density dependence is fish that spawn during the current peak in like May be fully saturated, right? But some of the most depleted life histories would be fish that enter earlier in the year. And so one way that we could get more fish back is potentially by expanding the number of life histories in the watershed, right? We need to recover depleted life histories because maybe we already have a peak, right? So we harvested these fish down to very low abundance. One of the things that happens when you harvest fish that are very diverse is you winnow away all the uncommon life histories and you end up with a much narrower range, and you just have the most common ones left, right? And so how do we get more fish? Well, we need to recover those depleted life histories. And so one of the challenges with Washington is we don't count any wild fish that come back before March 15th. Oftentimes we don't believe that they were ever there. There's pretty good evidence that they were, but regardless of what we believe and who we want to argue with, if we think about climate change that's coming and the world's getting warmer, it's unlikely that fish that spawn in May are going to significantly change their run timing over the next couple decades and start spawning in March or February, like we see fish doing in California and Oregon. And if we look at Seattle in 2080, it's predicted to be more like Fresno temperature-wise. Bellingham, I think, I looked this morning, it was like somewhere in Alabama. So significantly warmer. We may have a lot less snowpacks. We have a lot less to rely on in the spring. And so fish are going to need to spawn earlier. They're probably going to need to get out of the gravel earlier. And they'll probably mirror populations in California and Oregon more closely. And fortunately, like those fish are actually doing relatively well in a lot of places. We won't be outside of the range of what those fish can tolerate. But we need to have the life histories present to allow those fish to actually exist and occupy the habitat. And so that's where just managing for abundance and managing for current abundance and the current run timing is likely insufficient to provide a resilient steelhead population that's adaptable to climate change, and may underestimate the true capacity of the watershed. And when we have things like escapement goals, we don't generally have large enough escapements to truly test the capacity of the habitat and actually over-escape, as we would call it, and see what happens when we do put more fish onto the spawning grounds. And so just a quick conclusion. What the literature really strongly suggests, and this work suggests, is that density dependence is strongest during the first year of life, likely during the first summer. Sometimes it can be in the winter. Overwinter survival can be limiting. This is definitely not what's traditionally thought of in the Pacific Northwest in fisheries management. It really comes from the Atlantic salmon world. And it may not necessarily be true for any of the salmon species, right? This is one of the things that may be fairly different about steelhead that sets them apart and is very important in how we structure management plans. And again, this is occurring on these very localized scales, right? It's that because they don't disperse from reds early in life is why this difference occurs. And so, I don't mean to downplay habitat again, right? So if we know that fry stage is a bottleneck, we can actually target restoration and recovery actions at the fry stage to get more fish back, right? So we think about the Skagit, all my work's in the main stem. The main stem has been channelized. We've lost a lot of side channel habitat and complex habitat. So if you want to get more fry back, things like side channels and islands and increasing the amount of edge habitat could actually increase a lot of the numbers of fish that we get through the fry stage. And you create too much fry habitat, maybe then par habitat becomes limiting, but it's one of those things that we could try and focus on if we really do want to focus on steelhead in terms of recovery. And then all of our ESA recovery plans were supposed to manage for 4 VSP parameters, viable salmonid population parameters, which are abundance, productivity, spatial structure, and diversity. And currently, we only have ever come up with recovery goals for abundance and productivity because it's something that we can do where we have the number of fish and we run a stock recruit model and we say, okay, we think we should have this many fish and they should be this productive. And we have this pie in the sky goal that we never achieve. And one of the ways that we could get there is actually develop goals for spatial structure and diversity, which are tightly linked, right? It's Do we have enough fish throughout the range of spawn timing that exists? Do we have fish in all different parts of the habitat, right? And maybe not in any given year, maybe, you know, certain parts of the watershed will have good and bad years, right? But we could develop goals for how broadly distributed fish are in space and time. And a lot of this really just comes from GPSing reds. It's not like this is some huge ask that we would be making of people. We generally, it's very rare to actually GPS locate reds, even though red counts are the #1 way that we assess steelhead populations, their abundance. People just don't bring GPSs with them that much. So it's something that we could do that's a pretty low barrier to entry and start accounting for this. And so with that, that's the end of my main presentation. So the next chapter for me after I do my master's here is I'm going to go work with the conservation angler. So it's a very small nonprofit out of Oregon. I just wanted to give a little plug there. My other coworker there is John McMillan, who I know many of you know or have heard of. And it's a small group. We generally don't work on habitat because everybody else is working on habitat. There's a ton of great work out there. And so we have less to offer in that world. And so we really want to focus on the management realm of this, right? And where the main thing that we're focusing on there is essentially climate change, right? Is resilience and adaptation to climate change, which is sort of this fifth horseman, right? Heat. It's the fifth H that is not, it's talked about a lot, but it's not being addressed in terms of how our population is going to adapt to climate change. And so that's what we try to focus on. We're hopefully get some great new angler science stuff going. I forgot to mention Scott's work. So Scott and Bill McMillan, who's John's dad, have been doing some really cool work, red counts and getting GPS locations of those reds. And so time series of GPS locations of reds and tributaries is actually super valuable because it's something that we don't have. So we could actually combine that with some main stem data and also starting red counts earlier in the season to track patterns of how are we seeing fish come back earlier and earlier? Do we see variation year to year? Those kind of questions that we just can't necessarily answer without adding that kind of data. So getting more of those kind of programs and then just publishing good work and continuing to do work like this to try to improve the way that we manage steelhead through advocacy and occasionally lawsuits if we feel it's needed. So anyway, I encourage you to check us out at theconservationangler.org and donate there and everything. And I just want to say thank you to all my funders, Seattle City Light, NOAA Fisheries, UW, and also a big thank you to North Sound TU because you guys helped me throughout all this. I know at one point my funding was hung up for many different reasons, and you guys came through with a scholarship when I asked you for it, which was actually tremendously important because I was putting all that on my personal credit card, and and for helping and everything. And I just wanted to give a special thank you to you guys for that, and I'm happy to take questions.
Wild Steelheaders United, Trout Unlimited, encouraging us to get something started. It's really cool to see where it's gone. So.
Yeah, well, thanks. I'm glad to see you guys have run with it. That's pretty rare, so it's pretty cool. Yeah, there's a few familiar faces here. A little more gray. It's like me.
Time for questions. Nick, for the recording, if you'd mind, like repeating a short version of the question into the microphone.
Okay, I'll try to remember to do that. Yeah.
How are you accounting for density dependence with when you look at historical abundance? And I know that you basically we have to assume that the spring part of the run on the Skagit is probably at or near its capacity at sometimes with the dependency. But yet, we've lost the early part of the run or majority of the summer run, etc. I mean, that's where we have lost the numbers that would be in the watershed throughout the whole year.
Mm-hmm.
Sorry, I answered my own.
Okay.
It just doesn't seem to. I mean, we all hope that because we get more adults back and more spawn that we're going to get more fish back. But I know there's definitely a level of that. It just doesn't seem to me that with this habitat on the Skagit, say, being fairly productive and stable compared to other streams.
Mm-hmm.
that once produced 100 to 200,000 fish is now producing 5,000 fish, and we're at a density dependent level in certain areas of the stream. It just doesn't,
Yeah,
I'm sure there's more to find out. We can't ask them.
yeah. So I guess the question was, if we see density dependence in the Skagit, like how are we going to get more fish out of the watershed? Yeah, so I went back to this slide. I think I forgot to highlight what this blue box is telling us is this is the range of escapements for the years that we have red data for, right? So from 2,500 up to 9,000, so just over estimated capacity. And so then if we go all the way back to this plot, if I get rid of the high years up here where we're over capacity, we still see the exact same relationship, right? And so one thing is the evidence for density dependence when all we have is adult to adult data is it's pretty unclear where that is occurring in the watershed, right? We have 3 demographically independent populations in the Skagit, which are the Sauk, the main stem and tributaries, and then the Nookachams. And so it may be that where we're seeing density dependence in that overall curve, for one, it's pretty uncertain, but it may be from a place like the Sauk, right? Where we know that there's a lot of fish going back to the Sauk, and we still see density dependence here, but it may not mean that we're fully utilizing all that habitat, right? It's coming out of local clusters within small areas of the watershed because we're still seeing spatial expansion. So we could still be getting more recruitment, more adults coming back, even though we're filling up some areas. And then it's likely that it's possible that in lower river tributaries, like the Nookachams actually has quite a few fish in it, and they often spawn in like January, February. Some of those other lower river tributaries, for one, we're not, other than Bill and Scott, we're not really counting early in the season. So we may have more fish than we think, or those tributaries may be very depleted and we're just not really aware of what's going on down there, right? And so it's possible that some parts of the Skagit are not full while others are. And when all we do is look at adult to adult data, we're seeing a signal coming from the Sauk. And so that's one of the things that we don't really know where that's coming from. And because fry and juveniles from the Sauk are not swimming down and occupying all this other potentially unsuitable but unused habitat, we're still seeing density dependence. Does that help? Okay. Yeah, Scott?
Do you think we could better monitor the existence of that early component? Because early spawning is certainly some way to measure it. I mean, reds are very countable and visible, and they're there like on tributaries that Bill and I survey. We'll see them and account for that, but we often have winters like this past winter where it's cold for a prolonged period. So we're potentially seeing fish that may have entered the system in December, January, not spawning until the same time as fish that came up in mid-April and just got it done and work it out. It seems like if we want to focus on ensuring that we have a robust early component that's been depleted, got to be a way to account for what's coming into the system, and not necessarily spawning until the water temperatures dictate its time.
Yeah.
But so that we're protecting those fish. Early in the season is when all the directed harvest and angling is on most of our rivers around here still to this day.
Yeah.
Overlapping with hatchery returns.
Yeah. So the question was, how do we better monitor early fish, and not just early spawning, but early entering fish that may spawn later in the season? And steelhead are very hard to monitor, even adults, partly just because of the time of year they come in. I think one of the best ways is more expensive, but is sonar, right? And there's not a lot of overlap between winter steelhead and some of these other species of fish. The Skagit does have some pretty late coho, but you can distinguish them somewhat on the sonar readout just based on body shape. And there are AI tools that are getting better and better at that. And so that's what's used a lot in Alaska, and they distinguish species with sockeye and Chinook and everything coming in. And a river like the Skagit, you would probably need 2, and they're over $100,000 a piece, and you got to pay somebody to run them. But that's probably the best way to get at that, because otherwise, yeah, it's pretty tough. I don't expect a lot of people to be out there snorkeling and counting fish in January, and it's pretty tough in a main stem river anyway. So yeah, a combination of red counts, but sonar is probably one of the best methods to do that.
Can you answer about the status of the sonar that has been paid for and installed that is work? Isn't there one right now working?
Maybe there is in the Skagit.
That's for, put a sonar unit in the Skagit.
Yeah, they put it in the well, it's still in the well.
Yeah.
Okay. Okay. Yeah. Yeah. I don't know anything about that. I've been focused on this. Yeah. Okay. Who is doing that? Okay. Yeah. Because they ran one for a little bit on the hoe and then they stopped and then they have the unit, they own it, but I haven't heard about where it went. So maybe it's
Put it on the Samish and the Nooksack too.
okay.
For the
Yeah.
last-
Okay. Yeah.
You can have funding one year and then say it's gone when you had it last year.
Yeah.
Maybe don't have it this year because.
Yeah. Well, they also, the state had a big budget shortfall this year. So I think there was, a lot of strain on state budget. I'll have to ask because I haven't heard about that, but there's more and more interest all the time. I think that's where it's going. It might take us a little bit to get there. But as you say, to your point, where harvest is focused on that early run, I think that's really what happened historically. Was harvest was focused on the early run timing. They're the most depleted. Even small amounts of harvest could actually produce a relatively high harvest rate and prevent them from recovering. And there's not that many left. And so allowing them to recover is most likely where you'd see the greatest benefit in sheer abundance.
Those fish seem to be naturally going to be the most vulnerable because they're back in the river the longest. So
Yeah.
potentially out for 4 months. If that angling, those are the ones potentially getting caught repeatedly.
Yeah. Yeah. And they're on their way out during the fishery as well. So getting caught on the way out is also not great for them. They're more vulnerable then too. Yeah. Yeah.
Might be kind of off topic, but I'd like your opinion anyway.
Yeah.
When I read that when they took the Elwha Dam out, that the fisheries biologists were really surprised that a bunch of rainbows from these lakes and the headwaters up in the Olympic National Park were actually descending and then going to sea and coming back as steelhead. And they said that they weren't born in reds in the Elwha. And I just wondered if fish in Ross Lake or whatever rainbow have access to the Skagit, and could you increase your return rate in the, is it feasible that these lakes could be connected to the river in ways that they haven't historically?
Yeah. So the question is, could we see, I think, more production coming from smolts from upriver sources, right? Like we did in the Elwha once they pulled the dams out? Yeah.
Biologists observed at the Elwha was that these lakes where they've been putting in all kind of hatchery fish for years in these lakes, that the scientists couldn't figure out when they took the dam out, how did these fish that for 100 years had been blocked off, how did they know to go to sea?
Yeah.
They got huge returns, and they said that a lot of fish were coming that were just rainbows. That I don't know whether they were hatchery fish, but the numbers were a lot greater than what they had forecast.
Yeah, that is true. Yeah, they were surprised at how fast. I think everybody was surprised at how fast they came back, and they did come from resident rainbows. And so most likely, I got to snorkel it for 3 years and do the summer steelhead counts up there. And the first year we did it in 2017, it was by far the highest density of juvenile rainbows I've ever seen anywhere, of par, like 2-year-old fish. And my guess, and I think what's generally believed, is those fish were always producing some smolts, right? So because steelhead and rainbow are the same species, but they were never able to get back up in there. And so there was every once in a while maybe a summer steelhead that would return, or there was some, because they're mostly summer steelhead, right? These are coming out of the upper watershed. But they were never able to get back up there. And there are some smolts that are produced by rainbow trout don't survive very well. They don't necessarily have the right metabolism to go out and really take advantage of the ocean. And so offspring of steelhead are much more fit smolts. They survive much better. So I think what happened is you had the first few years that went out and actually survived and came back. And then once they were started getting tuned to becoming steelhead, then you had this very rapid recovery, like we would expect, the participation trophy thing, right? You get this, just everybody survives, you get this huge boost of fish that comes up and then they plateau. And so they went from 0 to 1,000 fish in like, I think 5 or 6 years, which was really surprising. I don't think anybody expected that. And it does seem that for whatever reason, resident rainbows seem to harbor the summer steelhead gene. They seem to be summer runs more so than winter runs. But we do see, going back to Skagit, we do see some Ross Lake genetics below the dam. And I think, I have to look back. I think that is in the steelhead population, not just the resident rainbow population, which is likely some fish dropping out of Ross Lake. And I don't know what the origin of those, if they're hatchery fish or whatnot. But there's been rainbows up there for thousands of years, right? So I'm sure there's always some trickle of downstream flow and hybridizing. I don't know that it's necessarily contributing a significant amount, but it could be. It's a huge watershed above the dam, right? So if you had a lot of smolts coming out, I mean, it could be. I kind of doubt, because they're resident rainbow smolts, that they would be that abundant and survive that well, but it's possible. But I think there's like one paper actually out of Western where somebody looked at that. It's not really well known as far as I know, unless they did more in the relicensing, which is possible. I just haven't heard about it. Yeah. Yeah.
You know, I look at some of the tributaries to the Skagit, you know, like Bacon Creek and Finney Creek and the Cascade and the Lavin. And those streams really, I mean, they're pretty good condition as far as, you know, the stream itself, riparian, not a lot of activity, not a lot of fish passage issues. And still on those streams, we don't see the populations coming up.
Yeah.
That's the part that baffles me on the Skagit. It's like, we have some really pristine habitat. I don't know what the occupation, or how well it's occupied as far as the fish. I know there's a lot of work done on Finney, but I don't know about the others.
Yeah.
You'd think that those would be strongholds, you know?
Yeah, I agree. I mean, so I actually did red counts and we did snorkel, bacon, and diopsid, and I don't have it in here. But we did look at those, and there are certainly not low numbers of fish in those creeks during these years. So 2022 was the main year of this study, which was a year we had about 200 reds. So it was a higher escapement year. I forget what it was, but in the upper Skagit, it was a pretty good year. I just broke things. No. I don't know. Well, good thing it's over. Yeah. But yeah. Yeah.
I fished it and it's like.
Yeah.
A fish pond.
Mm-hmm.
Fish on top of each other, all the way from the lake, all the way.
Yeah.
Down. And it's unlike anything, and it's not a joy for pictures, so no one goes see it. Not fun.
Yeah.
But I mean, the productivity,
Mm-hmm.
the density there,
Yeah.
would blow these numbers probably out of the water with. So, I mean, it's very odd. I know it's a low gradient stream.
Yeah. So I don't know what's going on with this thing.
I think we're fine.
Yeah. Good thing we got through it. So I don't know exactly why the CTEC has such high of density and other places don't. That's where I'm skeptical that density dependence is really super informative about capacity. I think it tends to always exist if you look for it, simply because of how they fill up the habitat, right? They're going to home in on the best places.
There has to be a density of everything.
Yeah, and you have to expand and fill out all that stuff. So that's where I think that, you know, I've never personally been to the CTEC. I've been to other places in Southeast and fished, and I think part of the challenge down here is we harvested fish for so long at really high rates. It takes a while for them to come back. And, you know, there's been a lot of work on this done in all sorts of fish species because we harvest all, you know, cod and everything. So what they generally see is that if you harvest them down to really, really low abundance, it takes many generations. So we've been harvesting them for over 100 years. And granted, maybe our harvest rates have declined. Maybe the rate hasn't declined as much as we would like. Certainly the number of fish has declined. But it's going to take them a while to come back and really re-express that. And in bacon and diopsid, I mean, there's a decent number of fish. I don't have it up here anymore, but the two that were sitting next to each other, those are Bacon Creek fish from March. Because in 2023, I went out and by the end of March, there was almost 30 reds in Bacon Creek in the lower 2 miles. And I have always heard there's a bunch of coho there that time of year, so I went and snorkeled. And they were all, I didn't see any coho. So as far as I know, they were all steelhead. But they're certainly not absent. I mean, they're hanging on. They seem to be doing well. So I just don't know where the upper limit is. I mean, I can't really answer that, right? Like, we have to wait and see what the fish can do. But that's where I think allowing them to, if we have a really big year, allow that year to be big, right? Don't harvest it down to the escapement goal. You know, you can probably still fish on them, but make sure that you're allowing them to sort of level up as they need to. And also, if we have an early year like we did in Bacon Creek that one year, those might be pretty important fish to survive, because oftentimes Bacon Creek has a peak in May, you know, and very few fish spawning in March. And so we might be kind of seeing a shift of early fish surviving better. And that's gonna be, you know, not every year, but yeah, I don't know. I can't answer why the SeaTech has so many fish and we don't. I just think it takes them a long time to rebound. And the SeaTech also, they actually went through, and I think in the 20s and 30s, there was a weir on there, and they would kill all the steelhead that came through. So it's certainly not that the SeaTech hasn't had harvest, but it's been many decades for them to come back, you know, and the eel and some of these other places were the same, where it collapsed in the mid-60s and then eventually came back when no one was paying attention to it. Maybe not to its former glory, but, you know. Yeah, back there, hit slightly first.
On how we end up from possibly 100, 200,000 fish in the river to carrying capacity.
Yeah. I mean, certainly the habitat is not as productive as it used to be. I don't expect that we'll ever get back to 100, 200,000 fish, right? I mean, I'm sure a lot of that, the lower river was probably very productive, very, you know, wide floodplain, tons of side channels, all sorts of stuff that's just not going to come back. So, I mean, I think it's a combination of everything that we've done to them. Climate change is also probably not helping. It's hard to say, but I think it really does come down to the H's for the most part, right? We harvest destroyed habitat. The hatchery seems to have had a negative effect. I mean, there's definitely some work on that. It's controversial, but it seems like that didn't help. I think potentially the main impact of that was just focusing harvest on that early run timing, right? And not clipping fish and just harvesting indiscriminately, you know? And so then, of course, there's some hybridization, that type of stuff that eliminates life histories. And so, yeah, it's just a death of 1,000 cuts, really. And I think that's the problem, is it's going to take a long time to rebuild.
That seemed to be the case on the Olympic Peninsula too. I mean, you look at a lot of those rivers, like Queets.
Yeah.
Almost the entire river corridor is national parks.
Yeah. Yeah.
The habitat hasn't really changed in 100 years. Still seeing that steep decline.
Yeah. And so in the 1950s on the Queets, for example, we harvested 5,000 steelhead in December, or 5,000 steelhead in December were harvested. And that's bigger than the most recent run sizes that we've had there, right? And so now there's very few wild fish returning in December. There's some, but it's really considered a spring fishery. And so we've seen on most of those rivers, run timing is actually like the peak run timing and peak entry timing is shifted about 2 months further into the spring, simply because we've lost that early component. And so, you know, maybe that makes the later component a little more productive because there's less fish to compete with, but overall, the watershed would be more productive if we had that full breadth of spawn timing, almost certainly. And partly because they would use different parts of the watershed. And even we see in artificial stream channel studies, and even with pink salmon, if you have even a month difference in spawn timing, you can actually fit more fish into the habitat during this early period of life, especially because they start to move offshore a little bit. So even though they only move maybe a meter out, that opens up that habitat near shore. And so you can actually fill in more fish on just these really fine scales. And so that by the end of the summer, they actually see more fry existing when they have short differences in emergence timing. Yeah. Yeah.
Make any sense to, since fish don't move very far from the red.
Yeah.
Does it make any sense to physically move them to a different place?
Possibly. I mean, so I don't think people have tried that with steelhead and they tend to swim back like they do that with hatchery fish, and they tend to just turn around and swim back to the hatchery. In the Elwha, they actually did that with coho because they were using the coho hatchery to jumpstart the run, and it wasn't really working. They're all swimming back to the hatchery, so they drove them around to streams for I think 3 years and dropped them off, and they stayed there and spawned there, and then that actually did jumpstart the run because it put them out into habitat. And so it's possible you could do that. I think that's really like in a risk of extinction scenario. Like if you're like on Hood Canal, right, where there's just very few fish left, you have a few, putting eggs out, taking fish in, putting eggs out, and putting them in different places can help. I think in the Skagit, they're relatively well distributed. If we had a good idea of what the spatial structure was and there were some places that really didn't have many fish, it might help to go put some fish there. That could help and kind of give them a boost, do it for a couple years. That's entirely possible. I don't think it's ever been done. We don't have a ton of success with that, but you know, a short-term boost could certainly help. Did somebody have a question? Yeah.
Just curious, what you see either in your data or the literature about any effect, flow regime has on kind of fry dispersal, because, the Skagit above the Sauk is a totally different river than the Sauk itself or the one below. And I know, one of the big points of contention in their licensing early on is, how do we measure the downstream kind of tailwater effects of the dams?
Yeah.
I'm just curious what you saw either in your sampling or what the literature suggests about, the dramatic ups and downs of a wild flow regime versus a heavily managed flow regime, what that might be doing.
Yeah. So we actually had an interesting year in 2022 when we were out there all summer, where we had really, really high flows through mid-July. And normally there's a number of fry out in June. And so we actually started our field season, I think, second week of June. And I was expecting to see fry already out in a lot of places. And we didn't see the first fry actually come out of a red in the main stem until July 19th, which was once the flows actually started to drop. I think they were like the second week of July, there's still like 15,000 CFS at Marble Mount or something like that. And so once they dropped below about 8,000, then we started seeing fry come out, and so it actually shortened our season quite a bit. We didn't get to collect as much data as we wanted, but it was sort of interesting because it didn't seem that fry were using that high flow period to distribute. But coho were out, Chinook were out, juvenile steelhead, like steelhead par were out. We did a lot of night snorkeling. So there was a lot of fish out. There was a lot of coho up in the bushes and the grass and everything. And they seemed to maybe be using it to sort of move. But it seemed like steelhead stayed in the gravel until it was sort of time for them to come out. And I think that's probably a temperature cue as much as anything, because it stayed really cold because it was snowmelt. But that's generally like brown trout and Atlantic salmon as well. They generally don't see them using high flow to move. And I think particularly for steelhead, they're spring spawners, and I think they're timed almost to come out right at the onset of like summer, or it seems like most of where we see really early spawning is usually in places like in Southern Oregon, where we have ephemeral streams, where they're going to come out sort of on the downslope of that hydrograph, or not be out during a really sort of whatever you call it, a period with a lot of fluctuating flows and freshets. That's what it seems like. I think that has to do with them being spring spawners and sort of how their juveniles exist. So no, we generally don't see them using that. Not super well studied, but yeah, that's my experience so far anyway. So, good question. Yeah.
So density dependent and marine derived nutrients, do you think there's a correlation
Yeah.
there? And the reason I thought about it is because his question about jumpstarting some of the things is one of my big pet peeves is, as somebody that works on culverts, and we know that it's gonna be a culvert that's coming out, and there hasn't been fish up above there for 70 years,
Mm-hmm.
that we don't actually do like my friend Dick Dolan, who actually helped put fish above the Elwha Dam to seed these things and get fish imprinted on there and actually start putting marine derived nutrients above these culverts that haven't had them for years.
Yeah, that is a great point that I didn't touch on, and it actually goes back to your question. So yeah, we have, not only do we not have as many steels views, so we don't even have as many salmon, and it provides a huge amount of food, right? So I think one of the most important factors of habitat, besides like just the right temperature and having some sort of cover, is just having food. Like, I mean, the highest density of juveniles that we have anywhere is in square concrete boxes and hatcheries, right? And it's because we dump a bunch of food in so they can exist there. It's not good habitat by any means. And so if we really want to recover steelhead too, I think that's another thing that we definitely need to do is we need to get more salmon back. Now the Skagit does have, I think, the largest run of Chinook in Puget Sound. There's quite a few summer Chinook that spawn in some of these places in Coho. So it's not that there's none. And so I don't know, I would imagine that if we did get them back more so, and it may be things like pinks and chum, we see a correlation between like a really big pink year and survival and growth of steelhead juveniles, you know? So it might be some of these other species too, getting them back to come back and spawn would really help. But coho are the ones that kind of go up into these tributaries too. And it seems like the fish, having natural fish go up and spawn is actually better even than putting carcasses out because they put their eggs in the gravel. And I think they also distribute themselves more broadly, but more nutrients seem to stay in the system and get taken up when we actually have higher escapements. So yeah, that's a great point.
Past an hour at this point, but have maybe time for one final question. Make sure that we give people enough time to get home. Okay, so let's thank Nick for his expertise and for his time.
Thank you guys for coming.